Okay, hello everyone. Thank you very much for joining our e-seminar series today with Professor Mahla Poudineh from University of Waterloo. Before going to her presentation, like always, I would like to go through some housekeeping notes. If you have any question during the presentation, please put your questions in the Ask a Question box on the bottom of your screen. And also there is a chat box if you want to introduce yourself, say hi, and where are you from, and talk to your peers. You can always chat in the chat box. So our next e-seminar will be Professor Mina Hurfar, Dean of Faculty of Engineering and Computer Science at University of Victoria. For getting the most updated information about the upcoming seminar series, you can follow us on Twitter with the hashtag TransLBME, or you can also follow us on LinkedIn, the organizer, Professor Akbari, and myself, to get this information on LinkedIn as well. So last but not least, we would like to thank our sponsor, Montreal Transmedic Institute, who sponsors these talks from the beginning of each seminar series that we started in July 2020, and this is the 79th e-seminar that we have today. So by that, I will introduce our speaker today. Professor Poudineh is an assistant professor at the University of Waterloo, Department of Electrical and Computer Engineering, and the founding director of the IDEATION Lab, Integrated Devices for Early Disease Awareness and Translational Application Laboratory, since January 2020. She received her PhD degree in electrical engineering from the University of Toronto in 2016. Prior to joining Waterloo, Professor Poudineh completed postdoctoral training at the University of Toronto and the Stanford University School of Medicine in 2018 and 2019, respectively. Her research interests include developing biosensing approaches for diagnostic and therapeutic purposes, continuous health monitoring, and translating biomedical devices to the clinic and market. Her research has been selected as Science Translational Medicine Editor's Choice article and highlighted in the Nature News and Views. She was the recipient of the Best Poster Award during the Micro and Nanotechnology for Medicine workshop held at the UCLA in July 2019. She has been also selected as an inaugural contributor for the Advanced Materials Rising Star Series and to the Nanoscale Emerging Investigator Team collections. By that, I would like to invite her to share her screen and start her presentation. Thank you so much for accepting our presentation, our invitation, Professor Poudineh.
Yeah, thank you, Homan and Mohsen, for the invitation. So I'm glad to hear and to share our works at IDEATION Lab with you and also the audiences. So I'm sharing my screen. So you can see my screen, right?
Yes. Now presentation. Yeah, perfect.
Great. Okay, great. Hello everyone, and thanks for joining me this afternoon. Today I'm going to talk about some of the recent projects and works that we are doing at the IDEATION Lab, which some of them have built on my previous work during PhD and postdoc. Okay, so controlling diseases and providing healthcare is one of the big challenges worldwide. So diseases such as cardiovascular disease, cancer, diabetes, and also Alzheimer's disease, and many others, they have been the leading cause of the deaths, even in the face of a worldwide pandemic that we recently went through it. So we need to test a vast number of people every day to be able to detect these diseases at the earliest stages. And after diagnosis and determining of the treatment, still we need to track the health status of the patients to make sure that the treatments are working effectively. So unfortunately, most of the current diagnostics and also healthcare tracking systems are complex, and they're suitable for practicing only in the central clinical laboratories. Certainly, if you had the misfortune of having a loved one in the intensive care unit, you know that there are like many tests that you would like to do on the spot to know what's going on. And it's the same for the emergency room, where we need to triage patients to be able to provide care to the ones that are at the urgent need. We need to have better technologies, because currently how it works is that after a sample is collected from the patient, it would have a long road ahead of it in terms of returning the test results. So doctors or nurse, they would collect the samples by withdrawing blood or swabbing the throat, and then the samples would be transferred to the uh, clinical laboratories. In the big labs, usually the sample would sit for a while, because most of these big labs, they would do the testing when they collect a bunch of the samples. The sample testing and analysis itself is a time-consuming and also expensive process. So there are different steps required. So sample purification, preparations, washing, and adding the detection agents. So it's like a very time-consuming process to do the testing. So when the testing is done, the results again should be transferred to the clinics, and the doctors or nurse, they would find the patient, they would find the patients, and they would and provide them with the results. And finally, at this stage, an action can be taken. So this can be a few days and sometimes a week after that a patient walks into the clinic or into the doctor's office. And this is a big challenge is one of the big challenge in the diagnostic medicine and healthcare. We need to have technologies that are suitable for practicing in this type of clinical settings, and not necessarily at the central laboratories. And these technologies even patients should be able to use them by themselves. So the question then is that, can we move testing from clinical laboratories to the point of care? And that's the question that I have been trying to address and answer over the past 10 years by developing new tools and biosensors that can enable rapid, sensitive, and point of care disease diagnosis. So a variety of biosensors exist that they interrogate different body fluids, such as blood, urine, or interstitial fluid, or ISF, which is a fluid that is present underneath of the skin, saliva, tear, or sweat. We have been specifically focused on developing biosensors for whole blood analysis and ISF analysis. Blood is the richest source of biomarkers. For example, in the blood of cancer patients, circulating cancer biomarkers can be found, or in the blood of a patient who is infected by bacteria, disease pathogen can be found, as well as like millions of DNA, protein, or small molecules that are in circulation, and they can be an indicator of disease and a biomarker of disease. ISF is also the second richest, and it has the most similar molecular compositions to that of blood plasma. At IDEATION Lab, to study whole blood and ISF, we are using two technologies. So we are using microneedles to extract ISF and study and analysis ISF, and we are using microfluidics for whole blood analysis. And today I'm going to share with you some of the works that we have done in this area, and I'm starting with our microneedle projects. Microneedle arrays, as the name says, they're like arrays of micrometer needles that they can penetrate through the skin, and they enable minimally invasive access to the interstitial fluid. So the legs of these needles can be adjusted in a way that they wouldn't reach the nerve. So that's why it's a painless technique, and it's a minimally invasive technique. So a variety of different type of microneedles exist, so such as solid, coated, or hollow microneedles. or hydrogel microneedles. And they have been used for both delivering of the drug and also for interstitial fluid extraction and analysis of the biomarkers of disease. At IDEATION Lab specifically, we have focused on hydrogel microneedles for sensing because of their several advantages. So they can be used for increased and rapid extractions of the interstitial fluid in their swollen needles, like in a small amount of time. They're highly biocompatible, and also their fabrication cost is very low. And that's why they're more economical for mass productions, and they can be widely available. So that makes them easier for translating into the clinic and market. At IDEATION Lab, we aim to integrate the sensing elements into the microneedles for applications of the sensing. This hydrogel microneedle has been used previously for both cosmetic and drug delivery application, and specifically in drug delivery, they have shown promising results. And currently, they are in clinical trials to seek FDA approval. But their application in sensing is more recent, and the existing work, they would extract ISF for off-site testing and analysis. Again, it requires laboratory and it's time-consuming. So we wanted to integrate the sensing elements into this microneedle array for in-situ and on-needle sensing that basically we won't need the laboratory for the measurement. And we aim to push the technology all the way to the market and in the clinic. So towards this goal, in one of our projects led by Hanjia, talented master's students in our group, and Amin, our collaborators, we developed a reagentless fluorescence microneedle assay for minimally invasive detection, and we call the technique RFMID. In RFMID technique, the recognition elements or the sensing elements are aptamer probes. So aptamers are single-stranded DNA or RNA molecule, and they're analogous to antibody and can be used for a specific target detection. So we have aptamer probes that is conjugated to a fluorophore molecule, and these aptamer molecules, this aptamer is hybridized to a displacement strand that is conjugated to a quencher molecule. In the absence of the target, the aptamer and the quencher strands keep the flow of fluorophore and quencher molecule in the close proximity, so no signal will be produced because the fluorescent signal will be quenched by this quencher molecule. But in the presence of the specific target, the affinity of the aptamer towards the target is more, and that's why the displacement strand will be basically kicked off, so it would release the quenching and we can see a fluorescent signal. So that's the sensing mechanism, and we link the aptamer displacement strand complex into the hydrogel network using a simple way, so we covalently link this complex. So this is then our sensing mechanism. So upon the target binding, we would be able to see a fluorescence signal. For fabrication of the RFMID, we use silicon molds. And in this slide, you can see the details. So these are like just the details, but the main point from this slide that I want to mention is that we have used hyaluronic acid or HA as the backbone of our hydrogel microneedle. This hyaluronic acid has been already commercialized and has been used in many cosmetic applications. And this would make it easier for us in future to translate the technology into the clinic and seek the FDA approval. So that was the main reason that we chose HA for our application. And also HA has like a very amazing swelling capability. So as a first, so in this slide, you can see our hydrogel microneedles based on this hyaluronic acid, how we fabricate them. So this is like the patch before penetration into the skin, and you can see the sharp needles. So at the bottom, you can see the sharp needles using SEM imaging. So these sharp needles are very important for effective penetration of the needles into the skin. Here we applied the patch into an agarose hydrogel that is loaded with the dye to be able to see if we can collect the ISF using our patch. So you can see that the patch absorbed the dye in their swollen needles, and also we were able to recover the dye effectively from our patches. So after the fabrication, as a first step, we studied the dysfunctionalization of the hydrogel microneedles with the aptamer and our sensing elements. So here we only link the aptamer probes into the hydrogel network, so we don't have the quencher strand. So that's why you can see a fluorescence signal. Next, we link the complex of the aptamer with the quencher strands into the hydrogel network, and as you can see, the fluorescence signal decreased. So we can see that we can effectively link the aptamer with the quencher complex into the hydrogel network. We used our platform, the RFMID, for detections of 4 different targets using their specific aptamer probes. So ATP is the molecule of the energy, glucose, all we know about it. L-tyrosinamide is a simple mimic of tyrosine, and it's like a simple amino acid, which is very important for catalytic actions of several enzymes, and it could be also biomarkers of the metabolic syndrome. And thrombin also is like a unique molecule that has applications that can be used as that is used as an anticoagulant. So we loaded, to mimic the actual in vivo experiments, we loaded the porcine skin with these biomolecules, and we applied the RFMID patch on the skin, and we measured the fluorescence intensity after the applications. So here is the results for the glucose measurements. As you can see, by increasing the glucose concentrations, the fluorescence signal increased. So it means like more of the quencher strands is released, and we're seeing like a higher fluorescence signal. So this is like the ex vivo study that we have done prior to an actual in vivo measurements. In the in vivo measurements, we used RFMID for measurements of the glucose in a diabetic model. So we had rats that are diabetic. We applied the patch into the skin of the rat, as you can see here, and we measured the fluorescence signal. After that, using our calibration curve, we defined the concentrations of the glucose. So the red graph is showing the RFMID measurements of the glucose in the diabetic rats. We also collected blood from the tails of the rat at each time point to compare the RFMID measurements. And as you can see, the measurements from the RFMID, the red graph, closely match the one from the standard glucose mirror. So it shows that the RFMID can be used for accurate and measurements of the biomarkers without the need for addition of any reagents into the into the assay upon the target capture. So 2 things that I want to point is that the RFMID can be used for detections of the targets, can detect the target within 2 minutes, and this comes because of our reagentless technique, since we don't need to add any detection agents upon the target capture, and also because of the swelling capability of our HA-based hydrogel that can extract higher amount of the ISF in their needles. So other microneedle diagnostic techniques that are using similar technologies, they require over 100 minutes for the target detection. In another work, we fabricated conductive hydrogel microneedle arrays that they can be used as the electrode. So in this work, we again use HA-based hydrogel, and we incorporated P.PSS into our hydrogel microneedle. So P.PSS is a conductive polymer. It can increase the conductivity of the patch. So in the bottom here, you can see the needles of our conductive hydrogel microneedle array that are again important for effective penetrations. And here is like the patch open after swelling, and you can see the swelled needles. So we use this conductive microneedle array as the working electrodes in an electrochemical sensor for two different applications. So in the electrochemical sensors, we do have the reference and counter electrodes, and we have the hydrogel microneedle based as the electrode, as the working electrode. In one project, Payman, an amazing PhD student in the lab, and Amin, our collaborator, integrated platinum nanoparticles into this conductive hydrogel microneedle for in-situ glucose detection. And in another project led by Sarah and undergraduate students in the group, we developed a hydrogel microneedle for pH measurement. So here we use the intrinsic characteristics of the functionalized HA for the pH measurement. Sarah worked on this project during her during her co-op term in our lab in the summer 2021, and because of her great work, she received the Co-op of the Year award at Waterloo. Unfortunately, she decided to continue her grad studies with us. So in these two works, we also look into the in vivo measurement. So we applied the our electrochemical sensor that consists of the 3 microneedle arrays into the rats, and we measure the glucose or the pH. So in future, we aim to combine the aptamers of the RFMID with this conductive hydrogel microneedle and develop platform for continuous and real-time measurements of the different type of biomarkers. So that brings me to the end of my talk on the our works on the microneedle projects, and now I want to switch to the works that we are doing with the microfluidics for the whole blood analysis. During my postdoc at Stanford at Tom Sauce group and in the collaborations with Kathleen from the Eric Apple groups, we developed a platform for continuous measurements of the insulin and glucose. The current approach for the measurements of the insulin in the laboratory is ELISA, and our goal was to miniaturize the ELISA into a microfluidic platform. So ELISA is a very sensitive assay, but it requires many washing steps and also adding the detection agent, so it's not at all a real-time assay. So our goal was to miniaturize ELISA into a microfluidic platform and also transform it into a real-time assay. So as all we know, the current gold standard for the glucose measurements are these glucometers. So these with these glucometers, diabetic patients or individuals, they need to prick their fingers, collect a droplet of the blood, and then use these devices for the glucose measurements. Continuous glucose monitoring or CGM devices have already revolutionized diabetes care, and diabetic patients, they don't need to punch their fingers every few hours. These devices, they can track continuously the level of the glucose within the interstitial fluid and provides the continuous measurements to the patient. Currently, or actually recently, these CGM devices have been integrated into the artificial pancreas device or APD systems for the automatic insulin administrations. The goal of this APD system is to actually mimic the action of the pancreas, which its action has been impaired in diabetic patients. In the APD system, the level of the glucose is continuously measured with the CGM. Then, based on some control algorithms that are based on the age, sex of the patient, the dosage of the insulin will be determined, and the insulin is injected into the patients using an insulin pump. So you can see that there is a gap here. Insulin is injected into the patients based on the level of the glucose, not based on its actual dynamics in the individual patients. And actually, that's a big problem, because if the level of the previously administered insulin is underestimated, there is a risk of giving excess of insulin into the patients. And this could lead to the acute hypoglycemia condition, which is a low blood glucose level conditions, and it could lead to the patients can go to the coma, and even they could die. So that's a really big problem. So to avoid such complications, the insulin should be given to the patients based on the dynamics of the insulin response in individual patients. In addition, it is well known that insulin response in different patients is different, and it depends on the kind of dynamics of the insulin in individual patients. So we do need to have personalized techniques to determine the dynamics of the insulin in the individual patients. So the question is that, can we measure other important diabetes biomarkers such as insulin in addition to the glucose in real time and continuously? So currently there isn't any way that can determine the amount of the insulin in the blood of the patients that are on board in real time. So we need to have such a techniques to avoid complications in diabetic patients. So to address this challenge, so that was a research motivation for us, and to address this challenge, we developed the RT ELISA device with the goal of miniaturizing the ELISA into a microfluidic platform for continuously measuring of the levels of the insulin in real time to determine the dynamics of the insulin in individual patients. Our RT ELISA is a bit-based assay, so that we have microbeads. So for insulin detections, we coated the surface of the insulin with the capture antibodies, and upon the target binding, we have the detection antibody, and a sandwich assay is formed on the surface of the bead. So we are using the same antibodies that has been used in the actual ELISA for detection of the insulin. For the glucose, since there is no antibodies available, so we have used the aptamer-based techniques, the same that I talked about in the RFMID project. So we linked the aptamer and the displacement strand with the fluorescence molecule into the surface of our microbeads. So this is our assay, and we incorporated this bead-based assay into the microfluidic device that consists of 3 main modules. And each of these modules basically mimics one step in the actual ELISA. So the mixing module mimics the incubation step of the reagents that are here, our beads should be mixed and incubated with the sample that contains insulin and glucose. And the depletion module basically mimics the washing step. So we are washing off all the blood components and unbound detection antibody in these washing steps. And after that, we would do the detection as the beads passing through the detection channel. The main components of RT ELISA, I can say that it's the mixing module, because inside this mixing module, the reaction, it happens. In the actual ELISA, the incubation usually it's like between several hours and sometimes overnight, and that's why it's not it's not a real-time assay. In the RT ELISA, to make it a real-time platform, we use the advantages of the microfluidics, and also we incorporated some herringbone structures on top of the channels, and we were able to decrease the incubation time from several hours to less than a minute for the target recognition. Since it was like a very important a step in RT ELISA, this mixing device, in a recent study led by Hamid, a great master students in our group, we developed a computational models to investigate this speed-based assay inside the microfluidic mixing. So Hamid developed a computational model, and he investigated different parameters that could affect the mixing performance, such as the size of the beads, the flow rates. And if you're interested, so I would definitely encourage you the paper that has been recently published in Analytical Chemistry. So this is our RT ELISA device. So it has the mixing device. It has the depletion where we are washing off the blood cells, and as the beads passing through these detection channels, we would measure the fluorescent signal to correlate with the concentrations of the glucose or insulin. So here is how we did the detection of these beads. So we use a spatial multiplexing. In the detection channels, on the top parts of the detection channels, we excite the glucose beads with the green laser. So we had the fluorophore molecule that can be excited with this red laser. And on the bottom, we excited the insulin beads with the green laser. So the glucose beads wouldn't be shine up in the bottom because there isn't any red laser here, and the insulin beads wouldn't be shine up on the top where the glucose beads are fluorescently labeled. So we have this spatially multiplexings to be able to measure glucose and insulin at the same time. And here is a video that I want to show you. Okay. So here is like the fluorescent speeds as they are passing through the detection channel. So as I said, on top we are seeing the glucose beads, and on the bottom we are seeing the insulin beads. So you may see that you may think that is the trace of one beads, but actually there are two different speeds. And we did several control experiments to make sure that, for example, the glucose beads are not like shown in the regions for the insulin beads. So we measure the fluorescent signals for these speeds, and we correlate these to these fluorescent signals to the concentrations of the glucose or insulin. So as a first step, we did some in vitro characterization. First, we have driven the calibration curve that we correlate the glucose concentrations or insulin concentrations to the fluorescent signal. Then we spike whole blood samples with different concentrations of the glucose and insulin, and we run it through the device. So here, each of these beads is showing the measurements extracted from one bead. So we measured the fluorescent signal, and using our calibration curve, we extracted the RT ELISA measurements. So, and on the top, you can see the actual spike concentration. So as you can see, the measurements extracted from the RT ELISA closely match the spike and actual concentrations. So having getting these results, these promising results, we continue to test the assay with the actual rats. So for these experiments, we have diabetic rats, and we withdraw blood continuously from the rat using a peristaltic pump, and we inject it into the microfluidic device. And here is like our detection channel that you can see as the beads are passing, we would be able to measure the fluorescence signal. And this is the results that we collected. So here, each of the blue dots is showing the measurements collected from the RT ELISA, and the orange squares is the measurements from the standard assay. So we collected blood from the tail of the rat every 5 minutes, and we used a glucometer for the glucose measurements, and also we performed actual ELISA to measure the insulin concentrations. So as you can see, the measurements extracted from the RT ELISA matches the one from this standard assay, but the resolutions that we got from the RT ELISA is much better compared to the standard assay, for example, for the insulin measurements. And these measurements now can be used to assess the dynamic of the insulins in individual patients. So currently we are working on extending this RT ELISA assay to add other important diabetes biomarkers such as glucagon and C-peptides, and have like a platform that can be used for measurements of all the biomarkers related to diabetes at once and determining their dynamics. So glucagon is also a hormone similar to insulin. It's used for adjusting the blood glucose, and C-peptide is co-secreted with the insulin from the pancreas, but it's cleared in a different pathway. So to be able to understand what is the dynamics of this molecule, it's going to be very important to have definitely better diabetes care, and also to address some fundamental questions in diabetes research. In another project related to the microfluidic and whole blood analysis, we developed an integrated microfluidic electrochemical assay for point-of-care cancer testing. So this project is led by Fatemeh, a great PhD student in the lab, and Nandini, a PDF in our group. So in this platform, we have two components: a plasma separators to collect and separate the plasma, and an electrochemical sensor for detections of the biomarker. So the most important things about this platform is that we do not need to have any pump for injections of the blood, and the plasma separation is happening because of the capillary force in the assay. So that's why it can be used for point-of-care testing. So we use the assay for detections of the HPV virus that is important for the cervical cancer detection. During my PhD, I also work on developing microfluidic-based assay for detections of and also profiling of single cells, single circulating cancer cells. And we're able to profile them based on their levels of the invasiveness. Currently at the IDEATION Lab, we are developing the next generations of these devices, and we aim to make devices that can be used for multiple isolation, and also profiling of different type of cancer-related biomarkers. Hopefully in future, I would be able to share more results on this ongoing project with you. So that brings me to the end of my presentation, and I hope that I was able to give you some insights about the works that we are doing for analysis of the ISF and whole blood using microneedle and microfluidic projects, with the goal of moving the testing from the clinical laboratories to the point of care. And at the end, I would like to thank my group at IDEATION Lab. So this picture is I think old for the last summer, so definitely we need to update it. And also I would like to thank my collaborators that over the past 2 years, I have been working with them, and also the funding agencies that supports the research at IDEATION Lab. and I'm happy to take any questions now. Shall I unshare my screen?
Yes, please. Thank you so much for your presentation, Mahla. Mohsen, we
Can
can't
you hear
hear
me
you
now?
very well.
Okay, all right.
Yes.
Thank you very much,
Yes,
Professor
perfect.
Poudineh, for your very nice and inspiring presentation. I really enjoyed it, and I'm
Thank
sure
you.
that participants also enjoyed your exciting projects and your presentation on these talks. So just while you're taking a few moments to rest, I'd like to remind everyone that our next speaker is Professor Mina Hourfar. She's the Dean of Faculty of Engineering at University of Victoria. So we are very happy to hosting her next week. Please make sure that you mark your calendars for her talk. Also, I'd like to again reiterate our appreciation from Transmed Tech for supporting these seminar series, which we've been running for almost 2 years. So without their help, we were not able to run the show. So thank you very much, Montreal Transmed Tech, for your support. So I encourage everyone to ask their questions using the Ask a Question box. This is a great opportunity to communicate with our speaker and then learn more about these innovative technologies that they're developing in their lab. So Reza has a question. First of all, is thanking you for your talk. Great talk. How do you determine the design requirements for the microfluidic in vitro platforms? Would you please make an example? So mainly the designs of the microfluidic devices. How do you come up with these designs? Maybe if you can explain it briefly, that would be great.
Yeah, I think one thing I would say in the microfluidics, so it depends on the applications that you are working with. So how, and like in the applications, for example, how much blood you need to, for example, in our work, how much blood you need to process. So if I can just give an examples about this RT-ELISA platform that I discussed. So the goal for us, we know that, for example, for insulin, ELISA is the gold standard, and we wanted to make it the ELISA basically transfer it into microfluidics. So we just say, okay, what are the different steps in the ELISA? One of the most important steps is going to be the incubation, as I said. And how to mimic incubations in the microfluidics, that would be the mixing device. So I would say first we just like what we did, it was like determining what are the steps and finding like the corresponding microfluidic techniques to be able to do the specific work that we needed. So, and like specifically for this RT-ELISA, when we come up with individual modules, the most important thing it would be to optimize their performance individually. So it has like different steps. So for example, for the mixing device, we first started with the buffer solutions, making sure that in the buffer our assay can work, and after that going into the whole blood, again just checking the individual device in the whole blood. So I didn't very talk about the depletion one. So the depletion one is like based on a deterministic lateral displacement, which is again is like a very well-known technique in microfluidic for sorting of the particles. So we use that for basically washing off the small blood cells and separating them from our beads that are larger than the blood cells. Again, we did the optimizations for the flow rates for each of these components. And when you want to do an integrated microfluidic platform, it would be very important now to adjust the flow rate and do some design considerations to be able to combine them together to make sure that their performance wouldn't be impaired. So again, like we did some simulations, adjusting the flow resistance to make it work. But what I would say in general, you need to know what is finding, like basically based on the applications, you need to tailor your design consideration.
Well, thank you very much. Great answer. So then I have a follow-up question from you. So these are the microfluidic devices that you prepared were designed for testing blood in mice. And we know that the amount of blood in mice is much smaller than the amount of blood in humans. So what do you envision to be the next generation of these devices for human applications?
Yeah. So first, I would say we test them in rats, not mice.
Rats.
Like mice, I guess mice has like 2 or 3 ml blood, but rat has like 25 ml. So it was more suitable for our application. So one thing I would say in this microfluidic one, we use our flow rate was 1 ml per hour. And to be able to assess the dynamics of the insulin, so we just need to do 2 or 3 hours measurement. So just imagine that it would be 2 or 3 ml blood from the patients. So it's just, I think it would compare to when you're going to the blood work and there you're giving like around 10 ml blood. So I think it can actually very suited for future clinical applications. And one of the important things about the RT ELISA technique that it's like a kind of ex vivo design. So you don't need to implant it. So actually that's why I would say that hopefully in future we would be able to translate it into the clinic much easier than other techniques that they need an implantable device. But we would need around 2 or 3 ml blood. And what I envision for this technique, it would be that the patients would come to the clinics and in the 3 hours, in the time frame of the 3 hours, we would continuously withdraw blood with them and adjust and determine the dynamics of, for example, insulin in the patients. And we can use these informations to adjust the dosage of the insulin for them.
Okay, cool. So, but the patients need to be in hospital settings to
that
be able...
would be for them. RT ELISA, they need to be in the because all the tubings and the pumpings. And actually, that was a kind of motivations for me to move to the hydrogel microneedles to be able to have a platform that can be used by patient outside of the clinic.
Okay, cool. So there is another question from Nahid. So thank you very much again for the great presentation. The question is about the CTC measurements that you had, or counting that you had. Can you please explain which properties are determining the invasiveness of CTCs?
Yeah, I think I just didn't talk about the circulating tumor cells a lot, so that was like my old PhD work, but definitely it's a very interesting project. So for the CTC, we use the EpCAM biomarkers that is on their surface, and these EpCAM biomarkers are only expressed on the cancer cells, not on the blood cells. So we had antibodies that are specific to this EpCAM on the surface of the cancer cells, and they're conjugated to the magnetic nanoparticles. So based on this labeling, we gave the CTC's magnetic properties, and then we use a magnetic force to pulling them down. But if you are interested, you can look into my paper in 2017 for more details.
Well, thank you. So the next question is from Behdat. So appreciate your generous presentation. So Behdat is curious about the microneedles you have developed. Are they disposable, or can they be used several times after swelling?
So currently we are thinking of that they would be disposable, and open the applications, they can remain, and after they are removed, so we are not putting them back. We specifically look for the lengths that they can be used in the skin for one of the devices, for the conductive hydrogel microneedles with the integrated platinum nanoparticles, and they can be used for 14 days without losing their performance. And so that's something that we are hopeful, definitely in future we aim to work on it to make them more durable. But if you are considering the cost of fabrication of this hydrogel microneedle, I would say having 14 days durability is very good compared to other, for example, continuous glucose monitoring patches that are available.
Okay, thank you very much. So I have a follow-up question about these. So these microneedles, they swell when you inject them into the, or no, sorry, not inject them. You puncture the skin with them, and then they penetrate into the skin. So when you want to remove them, is there any concerns regarding damaging the skin?
Yeah, that's a very good question. That all the reviewers are asking these questions from us also.
Who knows? Maybe I was one of the reviewers.
Yes, I know. So actually, this is very important characteristics of these hydrogel microneedles. As I said, they are minimally invasive techniques. And we have shown that upon removing of them from the skin, within 10 minutes, actually, the holes can be disappeared.
Okay.
So we did also several testing of using histology to make sure that there are not any inflammations from the microneedles. But no, they're minimally invasive techniques, and it has been shown that even it can be applied by patient themselves. So all these things, I would say, add them to their capabilities that hopefully in future they can be easily integrated into the clinics and market.
So how flexible are these microneedles that you're making?
So it depends on so the microneedles, the one that are based on only HA, so they're quite flexible. The one that, for example, we are adding P.PSS or other things, they could change. It can change their properties, but still they're flexible with the sharp needle. So the sharp needle is important for the penetration. Yeah, we are making sure that we are not losing the sharpness of the needles.
So they don't bend, the needles, they don't?
No, okay. They don't. They shouldn't bend. They should be very effective for the skin penetration.
So my next question is about the materials, because this HA base is photocross-linkable, so you probably have some methylated groups in it. Are there any concerns regarding the safety of these metallic groups? HA is by itself, it's biocompatible. And it has proven safety, but these new groups that you're adding and including the photoinitiator, do you have any concerns regarding the safety of these components?
Actually, so MEHA we are using exactly as you said. We did many histology experiments, again, to make sure that they are not damaging any inflammatory responses. And as I said, MEHA has been used a lot also for hydrogel microneedle fabrication, and other groups also have shown that they don't have the problem with the kind of the inflammatory or the other problems.
Great.
Yeah, we are making sure that we're using the biocompatible material, so it would make them easier for future clinical translation.
And how about aptamers? Are there any concerns regarding the release of aptamers or diffusion of aptamers into the?
Yeah. So what we are doing here, we are linking the aptamer covalently to the this hydrogel microneedle. And we have the quencher strand that is released, but there are some techniques that we are able to link them quencher strands to the aptamer that they wouldn't be released. And actually that could be used for real-time measurements also. We did histology again, and we haven't seen any problems with the aptamer molecules. But aptamer is this is a single and a short DNA strand, and they can be degraded very fast in the body. So we don't have any problems, I would say, with this aptamers. And also other groups have shown that they use the aptamers, similar techniques for in vivo measurements, but they put it in the vein for the measurements. So we are using ISF, but other people also have shown that they put it in the vein for the measurements, and they haven't reported any problems. So again, seems like they are short-stranded DNA, so they can degrade very fast, even if they are released. So we're trying to covalently link them so that the release amount would be less.
Okay, so but in terms of degradation, that you mentioned, they quickly degrade. So do you have any concerns regarding the degradation of aptamers when they are impregnated?
That's actually very important question. So what we are doing, so these aptamers, actually when we are using the hydrogel microneedles, then hydrogel network can use as a support for the these aptamer probes to make sure that it is like they're not degraded in the measurement. So what we did, so after like the first experiment that we did, it was in agarose hydrogel. So we used agarose as to mimic the skin, and then we did the skin experiments and we compared the results. So actually this experiment showed that, for example, that the aptamer wouldn't be degraded and wouldn't lose their performance when they're going to the skin environment. So I would say then our hypothesis is that the hydrogel acts as a support to help them.
Okay, cool. So I encourage the audience to ask questions. Otherwise, I will keep asking questions until Dr. Poudineh tells me to stop, ask me to stop.
No
or I get like humans, like, you know, stop. Like
maybe you can take a breath now without jumping.
Okay, you go ahead.
Okay. So thank you so much again for your very interesting presentation.
Thank you.
So I'm just curious about how far we are from clinical translation of these microneedles and what are the challenges that make it difficult to be translated, because it seems that the technology is really like cool and simple and easy to use. And since we are talking about translational biomedical engineering, I wanted to know this. Thank you.
Yeah, thanks. That's a great question. So one thing I would say, when we started the projects with the collaborators, we're thinking of, that's our goal, to translate the technology. And that's why I would say that we went through like kind of having HA-based hydrogel and also trying to make the technique as simple as possible, that when you want to go to all the FDA approvals, we won't have like that much of problems. But there are like definitely different steps. So we have started the animal works. Definitely we need to have like a larger animal works, like to make sure that these techniques are working in this large studies with rats, and after that large animals, so I mean like mini pigs. So these microneedles, the next step is always to go to a kind of a larger animal such as like mini pigs, since their skin is like more similar to the human. So we need to make sure that for example, in mini pig studies, we would get the same performance. And definitely there are steps that we need to work on it to make them more durable, as I was discussing, and like make sure that we do have like a good packaging to translate them. There are like some other things. So for example, like currently we are also working to miniaturize the electronics for our electrochemical biosensor, because we wanted to have the wearable platform. It should be miniaturized. The electronics should be also miniaturized. So I think like these are like these steps, and after that, definitely it would be clinical trials, which are like very important phases. So in one of the grant application that I had, so they asked us, okay, what is your timeline for translating these techniques to the market? And I was we came up with a 5 years actually timeline. So that's something that I think like could be reasonable, and that's something that we are working on it. So definitely our goal would be to translate the techniques into the market and clinic.
Yeah, hopefully. Is there any commercialized product in the market now or not?
So I know for the hydrogel one, the flu vaccine are currently in clinical trials, but I don't think that they're commercially available. So they're currently in clinical trials.
Thank you so much. So I think Mohsen is now ready to go for the second round. Please.
Well, I prepare the audience to ask questions if they have any, because again, this is a very exciting technology, and there are many interesting ideas being developed from this technology. Okay, so there is one question from Mustafa. So have you noticed any conformational changes or deformation of the aptamers in vivo, since their 3D structure is very unstable and sensitive to their environment?
So as I was answering Mohsen questions, so we're thinking of the hydrogel network as a support to help to make sure that the aptamer is stable. Another thing that I want to mention, it depends on also the process of the selections of the aptamer, that usually in the selection process, so that we are not working on them. But definitely the people who are experts in this area, so they're making sure that they would be stable in such an environment, in the kind of skin environment. So in the selection process, you need to incorporate some, for example, the actual environment, such as whatever is the DNA designs and all the things in this selection process, to make sure that they would be able to work in the for example, in the ISF environment where there are DNA designs available. So for example, one thing I want to say for our ATP work, so we observed that we are getting some reduce in the signal measurements, and that's probably because of the that ATP is not the aptamer very good for the environment. For the glucose, tyrosinamide, and thrombin, we didn't have that problem. So I think I hope that I was able to answer your question.
Thank you. So I have one last question, and I think we can end or conclude the session if there are no more questions. So my last question is about the sterilization method that you think can be used for human use, because that's a very important step towards clinical translation of this.
So this HA-based one, I think there are probably some UV or gamma irradiation I know that people are using for the microneedle patches for the sterilization. But yeah, definitely that would be one of the things that we should work on it for the future clinical translation. So I think UV or gamma irradiations can be used, but that's something that we should work on.
I'm mainly concerned about the aptamers that you have and their stability when you expose them to UV light or to high intensities of gamma or ethylene oxide, and then other types of established sterilization.
UV, so I don't know how long we need to have to do these irradiations for the sterilization, but for the actual for attachment, for the linkage of the aptamer to the hydrogen network, it happens through the cross-linking under the UV, and we didn't have problems with the degradations of the aptamer. But I definitely would depend on the durations that we need to do the sterilizations under the UV or gamma radiation. But that's one of the steps that we should work on it.
Awesome. That's great. Thank you very much, Professor Poudineh, again for your excellent presentation. I truly enjoyed it.
Thank you. And just one thing I want to mention is that we are looking for postdocs and motivated PhD students. Please reach out to me if you are interested in what we are doing.
Fantastic. That's great. And then also, I'd like to remind everyone that our next speaker is Professor Hurfar. And please follow us on Twitter. Our handle is TransLBME. You can also follow us, me and Dr. Savaji on Twitter and LinkedIn to get most up-to-date information about this seminar series. With that, I'd like to conclude this session and thank again our speaker and the participants for joining this meeting, and wish you all a great one.
Yeah, thank you, everyone.
Take care.
Thank you.
Bye.
Bye.
Thank you. Bye-bye.